Welcome back. So, in the previous lecture, I talked about isolation and characterization of DNA, and I just went over some basic concepts. So, this is the second lecture, and in today's lecture, I will go through the same topic, and I will focus mostly on the isolation of plasmid DNA from a bacterial source, because this is something that we will use in the next topic when we talk about recombinant DNA technology. So, just a brief recapitulation of what we learned in the previous lecture. This is, let us say, a bacterial cell which has both the genomic DNA and the plasmid DNA. The genomic DNA of the bacteria, let us say it is a E. coli cell, will be in the range of 5 million base pair long, and the plasmid DNA is again a circular DNA, but it is much smaller. It is only 6,000 base pairs long. Both are double stranded DNA. So, if you look at the cellular organization, this is how a bacterial cell looks, where everything is in the cytoplasm, and so it means that when we break open the cell, all of these cellular components, the DNA, the RNA that is synthesized, all the proteins that are there, all of these will come out into the cell lysate. Similarly, if we crack open a eukaryotic cell where we have even more compartmentalized components in the cell, but again when we crack open this cell, all of these will come out along with a lot of membranes, cellular membrane or different organelle membranes. So, we talked about DNA and RNA because this is one of the challenges that we face when we are purifying DNA, because DNA and RNA, they are chemically very similar. You will what we saw is that the sugar moiety is very similar except the H and OH at the 2' position. The nitrogenous bases are very similar except one nitrogenous base, which is T or thymidine in case of DNA and uracil in case of RNA. And the third component, phosphate group, is exactly the same. In case of both DNA and RNA, the linear chain is formed by these phosphodiester linkages between the 3' and the 5' positions in both DNA and RNA. So, that we have a 5' end and a 3' end. The only difference that we saw in terms of structure was that DNA forms a double helical structure, whereas RNA is single stranded and no such double helical structures are seen, but it can form other complicated structures. So, we also learned that RNA is chemically less stable compared to DNA because of the presence of this OH group at the 2' position. So, if the pH of the solution is high, then this hydroxyl group can attack this OH, resulting in a disruption of the phosphodiester linkage, so that this phosphodiester linkage is broken and the RNA chain is also broken. The same reaction can also be catalyzed by enzymes like RNase. So, there are different types of RNase, and we can use some of these enzymes to selectively degrade the RNA molecule present in our solution without degrading the DNA. So, this is something that we are going to exploit when we purify our plasmid DNA. So, there are various techniques of isolation of plasmid DNA, and they vary in different terms, but if you look at all these techniques, you can divide them into these 3 major phases, and we are going to use something that is very common in all these different techniques that are available. The first one is the growth of the bacterial cell, because we are going to isolate the plasmid from some bacterial cell. So, you have to grow the bacterial cell and then isolate the plasmid from there. So, once the bacterial cell is grown, you harvest it, and we typically do it by centrifugation, where the bacterial cells are precipitated out. We throw the media and then we work with that precipitated cells. Then we break open the cells to bring everything out into the solution, that is the lysis of the bacterial cell. And once everything is out into the solution, then comes the purification step, where we selectively purify, we are going to selectively purify the plasmid DNA. So, for this exercise we are going to use a common E. coli strain called DH5 alpha. We choose this strain for the extraction of plasmid DNA because this strain can maintain a high copy number of plasmids. Okay. So, the genomic DNA will have only one copy, but the plasmid DNA can have 50 or even 100 different copies in the same bacterial cell, which results in a very high yield of the plasmid DNA that you will recover from your purification step. In the next lecture, when we talk about recombinant DNA technology, we are going to again mostly talk about the DH5 alpha strain and the extraction of plasmid, and we will talk about what downstream experiments you can do from this purified DNA. When we talked about protein purification, we talked about a different type of E. coli strain, and that was BL21DE3 strain. So, in that case we used BL21DE3 strain because we wanted to make protein, and there are certain characteristic of that particular strain which are conducible for overexpression of the protein of interest that we want to use, so that we want to make more of our protein and then purify it from that particular strain. So, for proteins we use BL21D3, and for plasmid DNA we use DH5 alpha E. coli strain. So, once you have your bacterial strain, which is the DH5 alpha, and you have your plasmid which is inserted into that bacteria, you grow that in an overnight culture. This is something very similar to what we have done before during protein purification. The only difference is that in this case our cell culture is very small, it is only 3 to 5 ml in the Luria broth media. In case of protein expression, we used at least 1000 ml culture. In this case we are using 5 ml culture, and it is again grown at 37 degrees centigrade, and it can be grown overnight, so that you let it grow for several hours, 12 hours or 16 hours throughout the night, and you come back in the next morning and do your plasmid purification. So, you start your bacterial culture, let us say in the evening at 5 p.m., you let it grow, come back in the next morning at 9 in the lab. Now, you have your dense bacterial culture in this 5 ml media. You spin down the media in a centrifuge, where it will result in the precipitation of all the cells. So, that is, the cells are pelleted out, and you have a clump of cells at the bottom of your centrifuge tube. You throw the media from the top, and now you work on the cell that is precipitated out. So, what we have to do is we have to resuspend these cells. So, we do that using a buffer which is called a isotonic solution. So, what is an isotonic solution? Isotonic solution means that if you have two different solutions, and they will be called isotonic if the ionic strength of these two solutions are exactly the same. So, that if these two solutions are across a semi-permeable membrane, then you will not get much osmosis from one side to the other side. So, this is very important because the bacterial cell membrane acts like a semi-permeable membrane. We have intracellular components. So, there are solutions. So, it is basically a solution inside the bacteria, and the bacteria is suspended in some buffer which has some ionic strength. If the ionic strength of these two solutions, of the interior of the bacteria and the buffer that we are using, do not match, then there will be osmosis, which will result in either water flowing into the cells or flowing out of the cells, and both are detrimental because water flowing into the cells will disrupt the bacterial cell and it will result in lysis when we do not want lysis. Okay. We want to lyse the cell eventually, but we want to do it in a controlled manner. Okay. So, that is why we use an isotonic solution, so that we do not get lysis of the bacterial cell. This isotonic solution contains several things. One of them is EDTA, ethylene diamine tetraacetate. So, this is something that specifically binds different divalent ions. For example, it binds the magnesium ions and it reduces or it inactivates various nucleases which are present inside the cell. So, these nucleases can be DNases which can break down a DNA molecule, and we do not want that. So, to protect our DNA molecule, we add EDTA that will inactivate these nuclease enzymes in the bacterial cell. We also add from outside RNase enzyme. So, this enzyme is something that very specifically breaks down RNA molecule. As we saw that RNA is less stable, and this enzyme exploits that chemical reaction, that mechanism to break down RNA specifically. So, that will remove the RNA contamination from our sample. So, once we have resuspended our cell in the resuspension buffer, we add the lysis buffer. The lysis buffer has high salt concentration, and it is also highly alkaline in nature. Alkaline means the pH is very high. So, when we do that, it results in the disruption of the cell membrane, and this lysis buffer also contains sodium dodecyl sulfate or SDS. This is the same SDS that we use during SDS phage gel electrophoresis, and we know the structure of SDS. It has a charged head group and a very long hydrophobic tail. So, this is something that is very similar to the lipids in the cell membrane. So, it can easily disrupt the cell membrane structure, and once the cell membrane is disrupted, so now the cell membrane is disrupted, so everything that was inside now comes out into the solution. So, all the genomic DNA, the plasmid DNA, RNA, different proteins, they are all out in the solution. SDS will denature both proteins and DNA through hydrophobic interactions. The high pH of the lysis buffer disrupts the formation of any hydrogen bond. So, the double stranded DNA will also get denatured because of the disruption of the hydrogen bond. The presence of RNase A will chew up all the RNA that is present in the solution, thereby converting it into very small fragments or monomeric nucleotides. So, at this point we have a mixture of all these things in the solution. We have denatured genomic DNA, we have denatured plasmid DNA, and we have denatured protein. Now, what we do is we add another solution which is called a precipitation solution. This solution is a solution of potassium acetate. So, this is a salt, potassium salt of the acetic acid, and it brings the pH again to the acidic range. So, it brings up the pH to the acidic range. Now, since we are in the acidic range pH, you can again form the hydrogen bonds. The presence of dodecyl sulfate has already denatured the protein. So, the protein will not be able to refold and it precipitates. The potassium salt of SDS is not very soluble, so it also precipitates. Now, the major difference comes between the genomic DNA and the plasmid DNA, because chemically these two are very similar. So, this is where a proper experimental technique comes in. So, when we add this precipitation solution, what you will see is this solution turns white, because all this denatured protein and denatured genomic DNA, they will start precipitating out, and you will have to mix these two solutions, because initially we had the resuspension solution, then we had the alkaline lysis solution, and now we have the precipitation solution. So, you have to gently mix them, so that the genomic DNA, if you mix it very vigorously, then the mechanical agitation can actually start breaking the genomic DNA. So, we do not want that. We gently mix this. Now, the plasmid DNA, which is much smaller, can form the hydrogen bond. So, it can go back to its double stranded form and it remains soluble. But in case of genomic DNA, since it is so big, we saw that it is several million base pairs long, it is almost impossible to re-anneal, because it will not be able to find the necessary sequences to form the hydrogen bonds. So, the genomic DNA will not re-anneal and it will remain insoluble and it will precipitate out. If we use very vigorous mixing at this step, then what happens is the genomic DNA will start breaking down and it will form small fragments. Now, if the fragments are small, then they can easily find each other and become soluble. So, it is important to do this step very gently, where we have to mix the solutions without breaking the genomic DNA, so that it can precipitate out, and the plasmid DNA, which is because of its small size, can very efficiently re-anneal and remains soluble in the solution. So, at this step what we get is insoluble protein, insoluble STS, insoluble genomic DNA and soluble plasmid DNA. So, then again the microcentrifuge is put in a centrifuge and we spin it at a very high speed for a long time. So, we typically spin it for almost 10 minutes, so that all these insoluble components are precipitated out and the soluble plasmid DNA remains in the solution. So, the solution becomes clear, and in this clear supernatant we have the plasmid DNA, so that we can easily pipette it out from the top. This clear supernatant, which is taken out from the top, is then loaded onto a small silica based column, because this solution will contain digested nucleotides. It will contain several ions, and all these different ions that we have added, it might contain some of the small fragments of DNA and RNA. So, we have to remove all of them from the plasmid, so that we get a very pure DNA, plasmid DNA, at the end of our purification step. So, to do that, we add this supernatant onto a silica based membrane. Now, silica based membrane has negative charge, and this negative charge can interact with the positive ions that are on the surface of the double stranded DNA. This is done by using certain chaotropic ions, which can selectively facilitate this interaction between the plasmid DNA and the silica gel or silica membrane, so that the plasmid DNA sticks to this membrane, everything else is washed out. So, we do some washing step, just like we used washing step during protein purification using various columns. So, here we also wash out anything that does not bind the membrane. Our plasmid DNA binds to the membrane and it sticks there. Now, we have to elute out the plasmid DNA. So, we can do that by using a very non-ionic solution. So, we can use either water or we can use a buffer to elute out the plasmid DNA from the column. So, we also do this washing steps to remove ethanol, because ethanol is something that is used in some of the steps and it can be detrimental for the subsequent use of the plasmid DNA. As we will see in the next lecture, we are mostly going to use this DNA to do polymerase chain reaction, and there the ethanol can interfere with the polymerase chain reaction. So, it is important to remove any of these contaminants from the plasmid DNA, and that is done very efficiently by washing the silica column using wash buffers and then eluting it out using water or TE buffer. So, TE buffer is nothing but Tris EDTA buffer, which is at pH 8. Now, once we have this eluted solution of our plasmid DNA, we have to check its purity, and that is done using agarose gel electrophoresis. Remember that DNA is a highly negatively charged molecule. So, we do not have to add anything like SDS to make it negatively charged. So, once you add your DNA sample to the wells, and if this is the positive end, all the negatively charged DNA molecules will start moving towards the positive end. So, in case of a pure plasmid sample, we typically see several bands, if you are not going to see just one band. The reason we see several bands is not because of contamination, but different states of the plasmid DNA. So, this one, the one which is at the lowest at the bottom, is the one which is running the fastest, and this is a supercoiled DNA. So, remember, it is a circular DNA, and that circular DNA can again form more coils on top of that, resulting in a supercoil which makes its size highly condensed. So, since the size is small, it will run very fast, and that is what we see here. Some of the DNA molecules will get it will not be remain circular. So, it will become linear. So, since the linear DNA has a bigger size, it will run slowly compared to the supercoiled DNA. Even though it is the same sequence, same DNA, it will run slowly. And then the third one is where the circular DNA is highly relaxed and it runs even slower. So, we typically see such 3 bands for any plasmid DNA that we have purified, and this is quite acceptable. So, if you do not see any other bands here or here, and all of these bands are in the right place that you expect. So, you should also run a molecular weight ladder, and that will tell you that they are in the right molecular weight range. Then from that we can be very sure that our DNA is pure and we do not have much contamination from other genomic DNA. Once we have that, we will also have to check the absorbance at 260 and 280 nanometers. 260 nanometer absorbance will tell us the quantity of the DNA that we have purified, and the ratio of 260 to 280 will tell us whether we have protein or RNA contamination. So, if the ratio is very close to 1.8, we know there is not much protein contamination, and if it is not more than 2, so it is between 1.8 to 2, then we know that there is not much RNA contamination. So, we talked about plasmid DNA isolation, but in many cases we have to do a genomic DNA isolation also. So, when we typically do a genomic DNA isolation, we make sure that there is no plasmid present in that particular bacterial sample, because it will be very difficult to eliminate the plasmid DNA from the genomic DNA. So, again you start with same bacterial culture in LB media, then so you grow that overnight at 37 degrees centigrade, and in the next morning we pellet down the bacterial culture by centrifugation and discard the supernatant. So, the supernatant is the spent media, and our bacteria will be at the bottom forming a clump. Then again we resuspend the bacterial pellet in the lysis buffer, just like the plasmid purification, and break open the cell so that everything is now in the solution. Now, in this case what we do is a different type of extraction. So, we do, instead of using the column that we used last time, here we use phenol chloroform extraction. So, phenol chloroform extraction will result in different phase separation. The top layer will be the aqueous layer, which will mostly contain DNA, because DNA is highly negative and it will be very soluble in the aqueous phase. There will be a interphase, a white layer, which will contain mostly the denatured protein or even folded protein. And the lower organic phase will contain all the cellular debris, mostly the lipids, which are highly hydrophobic. So, now we can carefully remove the top aqueous phase, which contains the DNA, and it can also contain some other contaminants. So, we have to now purify the DNA from this, and it is done by selectively precipitating out DNA using cold ethanol. So, this aqueous solution is mixed with cold ethanol, and this ethanol is something that is chilled at -20°C and it is mixed. So, now the DNA starts forming precipitate. So, what we see is a white precipitate. We can centrifuge it at a high speed, so that all the DNA is precipitated, and then we remove the supernatant, because the supernatant will contain the contaminants and our DNA is forming a white precipitate at the bottom. That white precipitate is resuspended in 70% ethanol. So, this is a washing step. So, we can do it once or we can do it several times, so that we get highly pure white precipitate of DNA. So, once we have done that, we can resuspend the DNA in the same T buffer as before. We can check the purity of the DNA using an agarose gel, and we can determine its concentration using absorbance at A260, using the absorbance at 260 nanometer, and then we can store it at -20°C for downstream experiments. So, that is all for this week, and in the next week we will see what we are going to use with this purified genomic or plasmid DNA, how we use this DNA for recombinant DNA technology. Thank you.